BAP, shown at the top, is a carcinogen. It converts into BPDE, shown at bottom, via two intermediates. The final step of this four-mechanism pathway results in a DNA adduct with a molecule shown at bottom. Such adducts are the main cause of malignant lung tumors. BAP is found in smoke. It is a member of a large group of molecules known as polycyclic aromatic hydrocarbons. These molecules are the products of combustion of organic material such as soot, exhaust, ash, and cigarette smoke. BAP is hazardous to a vast amount of organisms, including humans. This widespread toxicity stems from BAP's ability to participate in base stacking interactions with DNA and is coupled by the interactions that BAP has with two separate enzymes found in the aryl hydrocarbon metabolic pathway of most organisms. These include a prevalent and diverse family of monooxygenases and a highly conserved hydroxylase. The family of monooxygenases, known as cytochrome P450s, place an epoxide ring onto carbon 7 and 8 of BAP. This forms BAP 7,8 epoxide. This epoxide ring is then opened by epoxide hydrolase to form a BAP 7,8 diol. A second epoxidation step yields BPDE, or benzoapyrene 7,8 diol 9,10 epoxide. Already capable of hydrophobic base stacking interactions with DNA, these enzyme-catalyzed transformations afford BAP with additional means by which to undermine the structure of DNA, notably the presence of an epoxide ring as a potent electrophile capable of forming covalent bonds without the need of a catalyst. Here, BAP is shown with the heme B molecule of P450's active site. Cytochrome P450, an unspecific monooxygenase, consistently attaches an epoxide ring to carbon 7 and 8 of benzoapyrene. This is due in part to the fact that BAP fits into the active site of P450 best when oriented with its pyrene group pointing away from P450's heme group. This is shown in further detail in the epoxidation of BAP section. We have proposed two mechanisms for the epoxidation of carbon 7 and carbon 8 of BAP, shown here. One utilizes a BAP radical cation and another two-step association of an electrophile and association of a nucleophile mechanism goes via a cationic intermediate. Both yield benzoapyrene 7,8 epoxide as a product. The molecule dissociates from P450 and enters the active site of the highly conserved enzyme, epoxide hydrolase. Through hydrogen bonding interactions between two tyrosine residues found in the active site of epoxide hydrolase, a general acid-base catalyzed process occurs to open the epoxide ring at carbon 8 and replace it with the oxygen of an aspartate residue. Again, two prevailing mechanisms are discussed. One suggests an SN2 attack by the aspartate oxygen on carbon 8 of the epoxide ring after protonation by a tyrosine, shown here, and the other provides for the opening of the epoxide ring without the assistance of the aspartate nucleophilic oxygen. This is known as an NIH shift mechanism. Both result in the opening of the epoxide ring at carbon 8 and the eventual formation of benzoapyrene 7,8 diol, shown here. After dissociation from epoxide hydrolase, benzoapyrene 7,8 diol undergoes a second epoxidation step by cytochrome P450 in the same manner as before, only this time at carbons 9 and 10. Once released from P450, BAP has taken on the form of BPDE, shown here, or benzoapyrene 7,8 dihydrodiol 9,10 epoxide. This molecule is now ready to form a covalent bond with DNA. Here, carbon 10 of BPDE is shown beginning to accept electrons from the N3 nitrogen of guanine after protonation of the epoxide ring. In this manner, BPDE covalently attaches itself to the guanine base of DNA.